The title of the journal that I'm going to introduce today is Proteolysis Activity Mapping and the Substrate Discovery Platform for Identifying Tumor-Activated Biosensors, which published on Nature Chemical Biology. Here are the highlights of this paper. This paper presents PSURF, a yeast surface display platform that directly screens protease cleavable peptide substrates in real tissue samples. PSURF captured context-dependent protease activity, including pH-sensitive cathepsin B substrate preferences and tumor-responsive sequences that were not explained by common benchmark proteases alone. Using differential selection against healthy tissues and tumor samples, the authors identified tumor-activated peptide substrates that distinguish metastatic lung lesions with higher specificity. One standout substrate, DNLG-RAF, outperformed conventional MMP responsive probes and showed that cleavage depended on precise sequence context rather than a simple motif. The study further translated these hits into imaging probes and nanobody-targeted urinary biosensors, demonstrating non-invasive tumor detection in vivo. Proteolysis is the biological process of breaking down proteins into smaller polypeptides or amino acids mediated by enzymes known as proteases. Proteolysis is now recognized for its highly advanced and diverse roles in post-translational modification and cell signaling. In a tumor microenvironment, dysregulated proteolysis is intricately involved in almost every hallmark of cancer, driving processes like cell growth, tissue remodeling, angiogenesis, invasion, and metastasis. Because of its catalytic nature and critical functional roles, capturing and modulating protease activity has become a cornerstone for developing next-generation cancer diagnostics and precision therapeutics. However, most existing methods either analyze purified recombinant proteases or map endogenous cleavage events, so they do not directly identify synthetic substrates that function in real tissue contexts. So this paper addresses that gap by developing PSURF, a yeast surface display platform that screens protease sensitivity peptide substrates directly in tissue samples and then uses the selected substrates to build tumor-activated imaging and urinary biosensors. This figure introduces the overall concept of this paper, which is to discover protease cleavable peptide substrates directly from real tissue environments rather than from purified enzymes alone. In this workflow, organs and tumor tissues are collected from animal models and extracellular fluid samples are isolated and applied to a large PSURF-based display library containing randomized peptide substrates. By subtracting sequences cleaved in normal tissues and enriching sequences cleaved in tumors, they identify substrates that report disease context proteolysis with higher specificity. These selected substrates are then taken forward into validation assays and engineered into functional probes for tumor imaging and urinary detection. The authors established how the PSURF library was built and validated before applying it to biological samples. They designed a yeast surface display system where each peptide is flanked by two epitope tags, V5 and HA, so when a protease cuts the displayed substrate, one tag is removed and the cleavage can be quantified by flow cytometry and sorting. To optimize this format, they tested several tag orientations using a TEV protease substrate and found that V5HA design gave the cleanest display and clearest cleavage-dependent signal change. They then showed that the platform can actually read substrate preference because Y-type TEV distinguishes the canonical substrate from a weaker non-canonical one, whereas a promiscuous TV mutant did not. Finally, they built a randomized 7-mer NNK library at very large scale, 10^6, and confirmed that the sequence distribution matches the expectation by NGS, and purified the uncleaved fraction to generate the final screening library used in the rest of the study. And then the authors used cathepsin B as a benchmark protease to validate the PSEF platform. They first show that under acidic conditions, PSEF enriches the expected LVG type motif, which supports that the screen can recover non-CTSP substrate preferences. They then show that as pH increases, cleavage decreases, and enriched motifs shift toward RR and VR, indicating that PSEF can capture context-dependent changes in substrate recognition. Importantly, they validate individual hits and identify RRLY-AFL as a new substrate that performs better than a conventional CTSP probe at pH 6.4. So for the overall paper, this figure is best viewed as method validation rather than the main translational result. In this section, the authors move from recombinant enzymes to the real biological problem, which is identifying substrates that respond selectively to tumor-associated proteolysis in complex tissue samples. They provide extracellular fractions from 2 long metastasis models as well as multiple healthy organs. And the first important observation was that many tissues share the cleavage of hydrophobic phenylalanine and leucine-rich sequences, indicating that simple positive selection against tumor samples would not be specific enough. To overcome this, they used a differential selection strategy. Starting from the tumor-cleaved population, they repeatedly depleted sequences that were also cleaved by healthy liver, lung, kidney, and the spleen, always carrying forward the non-cleaved pool. They then exposed this depleted pool back to tumor extracellular samples, so the final output was enriched for substrates that were spared by healthy tissues but sensitive to tumor-associated protease activity. NGS confirmed that the sequence composition shifted after this process, and from these enriched outputs, the authors chose 10 candidates and narrowed them to 4 tumor-preferred substrates for downstream validation. Here, the authors biochemically validated the PSEF hits using fluorogenic probes. All 4 probes showed faster cleavage in tumor lung samples than in healthy or inflamed controls, and they performed better than standard MMP-responsive probes. Interestingly, most of these substrates were not well cleaved by the common recombinant proteases typically used in probe design, which suggests that PSEF identified the substrates responding to more complex tissue-level protease networks. Further inhibitor studies and detailed analysis of DNLG-RAF showed that cleavage involved multiple protease families, depends on exact sequence context, and can occur at multiple sites. In this section, the authors tested whether a PSEF-derived substrate could be converted into a spatial imaging probe for metastasis. They engineered DNLG-RAF into a membrane-binding fluorogenic probe that becomes locally retained after proteolytic cleavage. After intravenous injection, the probes showed higher signal in CT26 metastasis-bearing lungs than in healthy lungs, and the signal correlated with the tumor burden. In ex vivo lung sections, fluorescence localized to metastatic nodules identified by histology and was about 2.2-fold higher in tumor regions than in adjacent healthy tissue. In the final results section, the authors translated the PSEF substrates into an in vivo diagnostic format. They built nanosensors in which each tumor-responsive peptide linker was attached to a targeting scaffold and a DNA barcode, so protease cleavage in the tumor would release a urinary reporter. After intravenous injection, the sensors accumulated in metastatic lung lesions and liberated DNA barcodes were filtered into the urine. The urine samples were then analyzed with CRISPR-Cas12a, which provided a sensitive readout of barcode release and therefore of tumor-associated protease activity. All four tested pieces of substrates generated significantly higher urinary signals in tumor-bearing mice than in healthy animals. So this section showed the translational endpoint of the study. Substrates discovered by tissue-level protease screening can be converted into multiplexed, non-invasive biosensors for disease classification. In this study, authors used a piece of directly screened protease cleavable substrates in real tissue samples, bridging protease activity profiling with functional probe discovery. Unlike recombinant enzyme or motif-based approaches, PCF captures integrated context-dependent proteolysis in the tumor microenvironment. The study moves beyond screening to translation, converting discovered substrates into fluorogenic probes, spatial imaging agents, and urinary nanosensors. The tumor-selective cleavage was driven by multi-protease networks and precise sequence context, not by one canonical protease or a simple consensus motif. However, the platform uses short 7-mer peptides, so exosite-dependent, structure-dependent, or longer-range substrate recognition may be underrepresented. In conclusion, PCF is a versatile discovery platform for tumor-activated diagnostics and potentially other conditionally activated therapeutic systems. This is the end of my presentation. Thank you for your listening.